Today, I'm excited to present an overview of Perturb-seq analysis. But before I start, I wanted to thank a few key people who have learned from over the years and who very generously provided time and content for this presentation. First, this talk is really to provide some intuition as to what a Perturb-seq dataset looks like and how to think about it and analyzing it in terms of the questions that we want to answer, and also motivate the kind of questions we can answer. Note that I'm using the term Perturb-seq to mean broadly parallel CRISPR single-cell screens. This talk will not be a deep step-by-step tutorial on the analysis itself, nor will we dive deep into the technical aspects or the experimental setup, designing guides, etc. But I do hope that you will have some ideas at the end of it to both think about new analysis methods and computational tools for Perturb-seq and similar screens. Finally, if you do have questions that are not covered in the talk, please feel free to email me, and I'm happy to connect you with experts on the more technical side if needed. With that, the idea of perturbations has really been central in molecular and systems biology to understand mechanisms and causality. So we may start with a system that we're interested in studying, where we can measure and observe a phenotype. So, for example, we can observe a system and measure it using genomic methods. For example, we could measure the level of RNA from each gene, which we call expression profiles. And we can do this for samples made of many cells, like a piece of tissue, or every individual single cell separately. We can then build a model of the system. This is the model of how we understand the system to work. We can go a step further and try to perturb or manipulate the components of this model, either the genetic or the non-genetic features. Once we perturb it, we can go back and try to observe the model again and measure how the phenotype has changed. Our observation of the system will now let us update what we know about the system and update our model, and then we can go on to perturb and so on. All of this is great. And traditionally, genetic perturbations have been well established for dissecting function. So in a classical perturbation scheme, as we see here, we can imagine that we have a hypothesis that a gene, here gene A, is associated with a certain phenotype. In this case, a change in cell composition. And we knock out the gene, and we do observe a change in cell composition. So here we see an increase in the number of purple cells or yellow cells, and a decrease in the number of green or blue cells. So we might conclude that gene A is linked with changes in cell composition. However, classic in vivo perturbation models that we think of in the lab, like a global or conditional gene knockout mouse model, they're really great for studying any system that we want, but they can be quite time-consuming to create. And usually, we study one gene at a time, and this becomes pretty hard to scale. On the other hand, now we have technologies like CRISPR, which have really allowed us to do arrayed or parallel CRISPR screens, which allow us to study many genetic perturbations at the same time, made possible because we can measure the abundance of each guide or perturbation at the end of the screen. Here, there are, again, a few limitations in that we would have to know what the readout is beforehand. So here it is quantifying the guides the composition of the guides in the cells that we have. And we would also need to know what the phenotype is. So typically, the phenotype tends to be cell growth or viability, et cetera. So in both the cases that we have discussed so far, traditional perturbation models, there are limitations. The limitations arise when we want to go a bit further and ask, what might be different molecular mechanisms that give rise to the same phenotype? What if we want to assess the cellular heterogeneity and ask how specific cell types or states are affected by the genetic perturbation? What if we want to predict regulators of multiple phenotypes at once? For example, we may want to see what state of the cell cycle a cell is in, but also understand how another cell circuit is reacting to the perturbation. We may want to also dissect complex biology for which we can't come up with a predefined readout, for example, cell differentiation. Finally, for a lot of the traditional perturbation experiments that I mentioned, we would have to do some sort of optimization for the readouts, selection, et cetera. And ideally, it would be great if we don't have to do so much prep and can perform a scalable screen. One solution is having a high content readout like single-cell RNA-seq or imaging, which really serves to bridge parallel screens like we discussed to complex phenotypes. Perturb-seq is exactly one such screen that aims to combine pooled CRISPR screens with high content single-cell profiling. Again, I use the term Perturb-seq to mean more broadly single-cell pooled CRISPR screens like crop-seq, mosaic-seq, et cetera. So let's talk about the Perturb-seq setup. On the one hand, we have pooled CRISPR screens, and on the other, we have single-cell RNA-seq. And let's start with a pooled CRISPR screen. Typically, we have a system of interest, and we have one or several hypotheses for which we have identified a set of target genes, usually on the order of hundreds or thousands. For example, they could be candidate genes from a GWAS study. For each of these target genes, usually we design 3 to 5 guides. In addition to the guides for the target genes, we also have control guides because we want to know what the perturbation did to the cells that we're studying in reference to a cell that did not get the perturbation. We need a system of delivery, usually a lentiviral vector. And finally, we want to grow the cells that have been transfected with a perturbation and then select them. So this covers the massive parallel perturbation part of it. The next part is the single-cell RNA-seq part of it. In the last few years, we have seen that single-cell RNA-seq has really transformed the way we study biology by giving us really high resolution. The key technical advance that allowed us to do that was to be able to identify individual cells with a cell identifier. Among the many different technologies, droplet-based single-cell technologies have really become very versatile and scalable and widely adopted. Briefly, there are oil droplets which have beads in them. These droplets capture cells in a microfluidic system. And within a bead, there are several oligonucleotide constructs which have cell identifiers. And we are going to go a little deeper into that. So here is one such oligonucleotide construct. To simplify the picture, we can think of this construct as having 3 key pieces. One is a sequence that is unique to every droplet. That's called a cell identifier, which identifies the cell that the droplet captures. A unique molecule identifier, which identifies each mRNA molecule in case of single-cell RNA-seq. And finally, a poly-T tail, which binds to the poly-A tail of an mRNA transcript. So now you can imagine an mRNA transcript that has a poly-A tail, a mature one, that then binds to this poly-T and is hence captured. And now if we can multiply our imagination to many, many transcripts and many, many such oligonucleotide constructs sticking out of the bead inside a droplet, then we capture a pretty big part of the transcriptome of the cell of interest. So to put things in context, here we have 3 droplets that have captured 3 different cells. We have a blue cell, an orange cell, and a green cell. And inside each of these, we have beads which have this oligonucleotide construct that we just talked about. And then we capture the mRNA from these cells and go through library prep and sequencing. So here we can see that each read inside each of these 3 cells has a cell identifier, a unique molecule identifier, and the transcript that it captures. So far, so good. So we have the pooled CRISPR screens and we have the single-cell RNA-seq setup. But how do we combine the two? So to combine the two, we really need to also have an identifier for the perturbation in each cell. And how do we do that? We have another identifier for the perturbation. So here's a schematic from the original Perturb-seq paper, which is a pretty complex and creative lentiviral construct. But the key take away from here is this guide barcode. In other words, to simplify it, we can think of another oligonucleotide construct, which has a sequence for either the guide, or there could be a guide identifier and another poly A tail. So with this in context, this poly A tail can now bind to the poly T tail of the droplet oligonucleotide construct. So to bring them all together, Perturb-seq identifies both transcripts and perturbations in each cell. So we have the cell identifier construct inside a droplet, but now the poly T tail can either bind to an mRNA transcript, or it can bind to a perturbation construct, which has a guide barcode. And with these two identifiers, we can identify both the cell and what perturbation is in each cell. So back to our Perturb-seq setup, we start with our CRISPR library, which has the perturbations of interest. Then we transfect the cells. Here, the term MOI stands for multiplicity of infection, which allows it to tune, on average, how many guides we want in a cell. Typically, for the purposes of this talk, we would like to have at least one guide, at most one guide in a cell, or one perturbation in a cell. And this is our system of interest. And then we have a selection process to make sure that we're only selecting cells that carry a guide, either a perturbation guide or a control guide. And then we go down to library construction. So based on what we saw before, again, this is subjected to droplet-based sequencing. So in the mRNA library, now, in addition to the droplet barcode or the cell identifier, we also have a feature barcode or a perturbation barcode, which identifies a perturbation. So we have the mRNA library. However, if you imagine the transcriptome of a cell, there are many, many more mRNA molecules than there are guide molecules. And so we perform an enrichment, usually using dial-out PCR, resulting in two different libraries. The mRNA library gives us one data set, which is our gene expression data set, as in any single-cell RNA-seq transcript. And the dial-out guide library actually gives us the perturbation matrix, which is a cell-by-guide matrix. And all of this pre-processing that can be done using standard workflows. If we were to have sequencing through the 10x Genomics setup, then 10x has its own pipeline called Cell Ranger, which has been adapted to CRISPR screens. Or we also have a cloud-based platform called Cumulus developed right here at the Broad, which has also been adapted to run on the cloud in a scalable way. To summarize our data sets now, we have an mRNA library, which gave us the gene expression matrix, which is a cell-by-gene matrix, where each element is the number of unique molecules identified of the specific gene transcript. And then we have a guide library, which gives us the perturbation matrix, where for each cell, we know the number of unique molecule identifiers that captured a guide sequence or a guide identifier. Now, at this stage, it's still a continuous valued matrix because you capture the number of unique molecule identifiers. But usually what we really care about is whether a cell has a certain guide or not. So we want to convert that into a binary matrix. And most of the standard software that I talked about have a way to estimate the background distribution, because a droplet can also capture ambient RNA, which is in the pool of cells that goes into the microfluidics. And so once the background distribution is estimated, we can then determine what the threshold would be to call a perturbation signal detected or not. Again, if you're interested to check out the cloud pipeline, which does all this pre-processing and returns these two matrices, then here is the link. And it was developed by Bo Li and Yiming Yang. Next, once we have the perturbation matrix, we want to do some sanity checks or quality controls to make sure that things look good. The first thing that we would like to know is the guide distribution. So if you recall, I said that there was this parameter called the multiplicity of infection, which can be tuned at the time of the experiment. This pretty much tries to tell you how many guides on average a cell has to have. So here, if we go back to the paradigm where I said that we want a cell to have at least a guide, we see that most cells here in the histogram have one guide, which is good. Next, we want to see what the cell distribution looks like. How many cells per guide do we see? This can be important depending on the question that you're interested in. For example, if you want to ask deep questions at the level of individual genes, then it's helpful to have more cells. But if you want to ask questions at the level of modules of genes or sets of genes or some patterns, then fewer cells per perturbation is good enough to give you a good signal. One of the last things you want to look at is whether a droplet captured just one cell or multiple cells. And now we have several tools available for detecting doublets, et cetera. Of course, we also want to know what the target gene actually looks like. If a cell received a perturbation, did the target gene, if the perturbation was a knockout, did the target gene expression go down? So measuring the target gene expression in relation to the control guide is one way of getting a sense for this. And finally, if a perturbation that you selected was essential to the system, then you will not be observing it because the cells probably would not have survived without it if it's a knockout screen. So with the setups all laid out, let's get to the more interesting part. What biology can we learn? So now we have two matrices. We have a gene expression matrix, which is a cell by gene matrix. And then we have a cell by perturbation binary matrix, which tells us if a cell has a perturbation or not. There are several questions that can get us from gene to function using the Perturb-seq setup. First, we may want to know what the cellular heterogeneity is in our system, simply because we have a high content single cell readout. Second, we want to know what are the transcriptional state changes that are driven by the perturbations. What are the cell circuits that changed? We have a high throughput expression profile as the readout. Then we may want to ask what perturbations have similar effects on transcription. These are also called co-functional perturbations or co-functional perturbation modules. Then we may want to know what are the effects of combinations of perturbations. So I said that for the purposes of this talk, we are assuming that the experiment has been optimized to have mostly one guide per cell, but you can imagine this scenario where there is more than one guide per cell. So how do these perturbations interact and what is the effect of their combination on the cell? And finally, we do have a single cell readout. So what are the cell type specific effects of the perturbation? So we're going to go step by step through all these different questions. So first, what is the cellular heterogeneity in our system? For this, we start with the cell by gene expression matrix. And we can use standard single cell RNA-seq analysis approaches to get cell types or cell states. So one standard approach would be to derive highly variable features or genes from our expression matrix, embed the matrix in a lower dimension, and then perform graph-based clustering to get clusters of cells, which might represent a cell type or a cell state. So as an example, there was an in vivo Perturb-seq study that was published recently, which aimed to look at the effect of genes implicated in autism through a GWAS study on brain development in mice. In this case, there are multiple cell types and 5 cell types were found. But in addition to the cell types, we can go deeper into each cell type and do subclustering to identify cell states. So this is something that any single cell RNA-seq experiment can give us, and so can Perturb-seq because of the readout. Now we can ask, what is the cell composition across the different perturbations? Are there some perturbations which favor some cell types over the other just because of their effects on transcription? So here's another example. Here's the example from the same study where for these 5 cell types, we can have a look at how the composition varies across the perturbation genes. Next, we can ask how many and which cell states were found. So here's an example where for the same cell type, there are 3 different states. 2 of them are activated. And within the 3 states, there are cells in the different stages of the cell cycle. So these are typical questions that come up in a single cell study in terms of the heterogeneity of cell types and cell states. Then we can ask, what are the transcriptional changes driven by the perturbations? This question can be further broken down to ask, what does each perturbation do to each gene in the cell? We can think of this as comparing cells with perturbation to cells with the control guides or control cells to ask what is different in the cells which have the perturbation. This is a very typical analysis setup for differential gene expression analysis. So if we can think of differential gene expression analysis, we want to compare expression of genes between 2 conditions. Here it is the cells with perturbation and without perturbation. And once we have the set of differentially expressed genes, we can functionally annotate them using the many different tools that we are familiar with. One approach that has been very successful is a regression-based framework that was first introduced in the Perturb-seq paper called MIMOSCA. So here, in this regression framework, the outcome variables here is the gene expression matrix, which we already identified before. And the design matrix has the perturbation matrix. This is the binary matrix. And the regression framework offers the advantage of adding other covariates that we may want to correct for. For example, there might be a batch effect in our data that we want to include as a covariate, or we may want to include some other cell quality metric as a covariate. And finally, what we're really interested in is in inferring this coefficient or the regulatory matrix. This coefficient or regulatory matrix gives us the effect size or the effect of each perturbation on the expression of each gene. A side note is that sometimes a cell may have a guide, but not actually get perturbed. We will not be going in depth into that today, but I just wanted to make a note that and refer you to the Perturb-seq paper, where they came up with a strategy to update the perturbation matrix, the binary matrix, based on a certain score that they found. Another approach is called mixscape, where they call some cells as escaping cells. That is, they do have the guide, but for various reasons, as the system is not perfect, they do not get perturbed or don't show the perturbation. With that note aside, let's get back to our system. We are in this regression framework, and we are trying to infer the coefficient or regulatory matrix. We can visualize this as a gene by perturbation matrix, where each element is the effect of the perturbation on the gene. One common way of visualizing it is a heat map. So here we have every element signifies the effect of a guide on the x-axis, on the genes on the y-axis. Here, the red color implies that the gene expression increased on perturbation, and the blue color implies that the gene expression reduced on perturbation. So this way, we know at a high level what the effect of each perturbation was on each gene. But you can also start seeing that there are some patterns. There are some guides which seem to affect genes in a similar way, which brings us to our next question. Can perturbations have similar effects? Perturbations which have similar effects are also called co-functional perturbation modules. And by going back to this matrix of guide effect sizes, we can see that there are these blocks or modules where a guide has a similar effect on a set of genes. And these are called co-functional modules. And these may be perturbations that act and have a similar effect are maybe acting together in some way. The next observation from this matrix is also that there are some genes which get affected in a similar way by certain perturbations. And these are called gene expression programs. So with having defined what co-functional perturbation modules are and gene expression programs are, we can then think of other ways to visualize them. So the network that we see here is one way in which the relationship between modules and gene programs can be visualized. In this case, the red and blue color imply inhibition or activation, respectively. And the terms in the program boxes are the functional annotations of the sets of genes that go together. Another way to view the same a subset of this result is using a network where we could pick out the target genes, which are transcription factors, and try to see the relationship between them, how they regulate each other. So far, we have tried to ask what is the effect of every perturbation on every gene, but we can also ask a different question. Sometimes it might be interesting to know the effect of a perturbation on a set of genes or a pattern of gene expression. So we could use the same regression framework, but instead of on the gene expression, it would be on some score of a set of genes, which might be either a gene program inferred by topic modeling or matrix factorization or weighted gene correlation network analysis. It might also be a low-dimensional embedding. So then we are left with a matrix, which instead of gene by perturbation would be module or embedding by perturbation. And this was done in the in vivo Perturb-seq paper that we've been using as a case study all along. So in that paper, for each cell cluster that was identified, gene modules were identified, and gene modules were paired to the specific cell type. And then the effect of each perturbation on the score of the gene module on a cell was determined. So this gives a slightly different representation. Here we see, here each row represents the effect of every perturbation on one specific gene module. So PN1, PN2, they're gene modules. And what's interesting is that some of the gene modules here are cell type specific. So we do see that there are some perturbations which impact some modules in a cell type specific manner. Besides Mimosa, there are a couple other tools that I just wanted to share, which also use similar concepts. So there's one method called MPG, which uses a topic modeling framework to get at the same question of how do we detect the effect of a perturbation on a set of genes or a gene program. And scMagic has a regression-based framework trying to get at the same question. So going on with the question of which perturbations are similar to another concise representation of it is using a correlation matrix. In this correlation matrix, each element is the correlation between the effect size vectors of two guides. So if two guides are impacting genes in a similar way, we expect them to be highly correlated. And imagine if you have a really large screen or a genome-wide screen. This gives a very concise and high-level view of the different modules that we possibly expect to see in the system and what to go after in deeper study. And the histogram on the right shows the histogram of the correlation coefficients between the effect size vectors of every guide. And as expected, the correlation coefficients between guides within a gene are higher than the correlation coefficient across the genes. And of course, because of the single cell readout and the sparsity, the correlation coefficients are not as high as you would expect. Which brings us to the next question, which is, what are the possible cell type specific effects of perturbations? We delved on some of this before, where we talked about the modules, and we saw that the effect of the modules were specific to certain cell types. So in the same example that we've been using so far, which is the in vivo Perturb-seq study, where we want to study the effect of certain autism-related genes on brain development, we saw that there were certain genes that affected certain modules in a cell type specific way. So for example, here is a PN1 module. So this is a gene module where represented by these genes, which is specific to the PN cluster of excitatory neurons. And it is also significantly enriched in that cluster. So similarly, there are specific gene modules which are impacted in a cell type specific way, which gives us a lot more information that would not be possible in a non-Perturb-seq styled screen. So finally, we ask, what are the effects of combinations of perturbations? Within the regression framework that we've been going along so far, this can be added as an interaction term. So we can imagine adding an interaction term for combinations of perturbations that are detected within cells. So again, the setup is similar. We have a gene expression matrix, and then we have the design matrix, which is binary, which is the perturbation matrix. And then we want to infer the coefficients or the regulatory matrix. The only difference is that we also want to know what the effect sizes of combinations or interactions of perturbations are. This can help us understand how perturbations act in relation to each other. Are the effects just additive without any interactions, or can they be synergistic, etc.? And so here is one way to visualize the effect of combinations of perturbations as seen in the original Perturb-seq paper, where interestingly, certain combinations of perturbations were enriched in certain cell types or cell states. So the numbers 0 through 6 are the different cell states that come out of clustering your single-cell RNA-seq dataset. And we do see that the effect sizes of certain combinations of perturbations are enriched in certain cell states. So with that, I want to summarize what we've seen so far. So we learned that Perturb-seq tries to combine pooled CRISPR-based screens with high-content single-cell profiling. And we also walked through some of the biological questions that such a screen helps us answer. We learned about co-functional perturbation modules, expression programs, and how we may visualize them and how we may connect them, and also the cell type specificities of these programs, and finally, how combinations of perturbations work. So with that introduction in place, we might want to scale our Perturb-seq study and want to scale up to many, many cells and many samples or mice, etc. So cell hashing has been commonly used to scale single-cell RNA-seq, and we can do the same in the context of a Perturb-seq study. So cell hashing usually involves overloading the channels or the microfluidic channels, and later inferring if a droplet captured a singlet or a single cell or a multiplet or multiple cells, etc. The slight variation from what we saw before is that in addition to the mRNA library here and the dial-out or enriched guide library, we also have an enriched library of the antibody hashtags. So antibody hashtags are often used for cell hashing. So this results in 3 data sets now. We have the cell by gene expression matrix. Now the antibody hashtag actually gives us a cell by antibody hashtag matrix. And finally, we have the perturbation matrix, which is the cell by guide. Now the cell by antibody hashtag matrix is what we would have to deconvolve to infer if a cell is a singlet or a multiplet, etc. Again, the standard software that are used for pre-processing are set up to handle such data sets and return 3 different data sets. The key step in terms of the hashing is to infer whether a cell is a singlet or a multiplet. And there is an algorithm called DMUX-EM, which is again part of the Cumulus suite, which can do this for you. Knowing whether a droplet captured a singlet or a multiplet or unknown allows us to add an additional layer of QC to our original RNA-seq data set and only focus on cells that we are confident are singlets. On a related note, going back to the question of how many cells may we need or how deep should our sequencing be, again, that would depend on the biological question that we're interested in. If we are interested in probing individual genes, then we would need many more cells. On the other hand, if we want to probe signatures or programs, then fewer cells per perturbation are sufficient. So in summary, we've discussed a functional genomic suite for biological discovery used in the context of single-cell RNA-seq. But it doesn't have Perturb-seq, the idea of Perturb-seq does not have to be limited to single-cell transcriptomics. And one can imagine adding other information-rich single-cell readouts at the protein level, at the chromatin level, et cetera. One such example is Perturb-CITE-Seq, which is a multimodal genetic screen, which tries to combine Perturb-seq and CITE-Seq. CITE-Seq is a technology which captures cell surface proteins using antibodies. So Perturb-CITE-Seq has an additional step of staining with CITE-Seq antibodies, followed by single-cell RNA-seq, such that we get both protein and RNA readouts. Of course, we can't profile the entire proteome. It's usually a small set of predetermined cell surface proteins of interest. So if we think of our setup in terms of co-functional modules and programs, we can do a similar application analysis of the Perturb-CITE-Seq, except that now we are going to know the effect of the target genes or perturbation on both RNA and protein features. If you're interested, the Perturb-CITE-Seq paper is also published and would make for a good read. Where would you go next if you wanted to delve deeper into Perturb-seq and play with some data sets, come up with new computational methods? A wonderful review was published just this week, and I'm just showing a table from it where they have summarized all the different perturbation modeling approaches in single-cell screens that have been published to date. Today, we discussed the first set of more classification and regression-based schemes. We also talked about factor modeling or gene module-based modeling. There are nonlinear modeling techniques out there, which we could not go into today, but might be worth a read. In addition to this resource of modeling approaches, you may want to play with publicly available perturbation data sets to come up with new models. And this review offers that as well. With that, I hope this talk gave some overview of what Perturb-seq technologies are and what applications might be, and I'm happy to take any questions.
Thanks so much, Ayshwarya, for that really wonderful and thought-provoking, and also I will say extremely clear talk. So thank you so much. We have changed our logistics a little bit. Sarah is kindly agreeing to voice any of the questions that come up on the Q&A, but I think I'll get started just by asking you a couple that I saw and I've been thinking about myself. One of them was a very practical one, which is that one of the audience members asked if you could perhaps just redisplay the slide that would allow people to find Cumulus online if they want to get to that. It seemed like there was a lot of excitement about using that as a tool.
Sure.
And I have a bunch of questions because I found this so exciting, but I will try to restrain myself and just ask one to start, which is that I was really interested. Sorry, I'll let you get back to the slide first. I'll try to restrain myself.
Okay. Please go ahead.
Great. Yeah. So I was really interested in this point that you raised about, and it was sort of a theme throughout, about combinatorial guides, possibly going into a single cell. And it seems like that ties in nicely with an idea you raised before about being able also to screen whether there are any perturbations that are just not consistent with cell viability, basically. And if I can imagine, and there may be reasons that this is technically not possible, but I can imagine that if there's a case or a bunch of instances of a case where multiple guides go into the same cell, you might also be able to ask whether there are particular combinations of perturbations that are not consistent with cell viability. And I'm just curious about whether the system allows sufficient confidence in the independent entry of guides that you can see particular combinations that are diminished in their abundance such that you can make that type of inference.
That's a really good question. I don't think I know the full answer to this. I'm happy to talk to some of my experimental colleagues who have played more with the tuning.
Sure.
I do know that in general, cells are not happy when you go for more than four guides in a cell. They don't survive as a rule. But it is an interesting question if there are certain combinations of guides too, which can affect the viability.
Yeah, no, it's a really interesting point. I think it's of course a persistent challenge how much of it is the guide is compared to what it does. That's great. I have, as I said, a bunch of other questions, but Sarah, I want to make sure that you are able and have space and time to voice those from the audience. So
thank you. There's one in the Q&A box from Dr. Flores who says, it's just a wonderful compliment. Thank you. This is extremely helpful. Slides and presentation were fantastic.
Thank you.
One of the questions I had was just regarding cell type for Perturb-seq and whether you've observed that certain cell types are more amenable to the process or others have been more difficult to model due to logistical or just heterogeneity in the actual cell populations.
So that's an excellent question. Again, maybe more for an experimentalist who actually handles the different cell types and who might be optimizing the protocols. I don't want to guess, but I would say that a lot of these studies have been done in vitro. So doing an in vivo Perturb-seq is definitely more challenging. Also, some of the earlier studies have worked with immune cells or cells that are very comfortably grown outside. So I would imagine that we may run into the same challenges that we run into in terms of even single cell RNA-seq for complex tissues. But for more details, I would have to defer to my experimental colleagues who do the hands-on work on that. But it's a great question. Yeah.
Sarah, are there other Q&A questions we should address or?
That is all for right now. So if you had more questions, Anne, that'd be perfect.
I will then maybe perhaps end with a very far-reaching question, and it's just a speculative thing in my mind. But I'm really interested in taking the word perturb into the broader sense and thinking about perturbations to the environment that a cell finds itself in. And I wonder, so for example, I'm currently embarking upon a new study where we're exposing cells from different species, and I think you and I have discussed this in brief before, but exposing cells from different species to a bunch of different temperatures to see how they respond. And I'm curious about whether you would advise us, if this is a promising or a really bad idea, to try to apply the same sort of analytic framework that you and others have developed here to try to think about perturbation in the broader sense, where it's not a guide-mediated perturbation on the level of an individual part of the genome, but an environmental effector, basically. And I wonder if we could use some of the downstream analysis that you've developed to ask about how the environment matters and what its specific effects are.
Yeah, I think, again, I'm not an expert in that, but I think we could imagine if we had controls not in the environment and observations in the environment, we could have a setup like we've discussed in terms of the analyses to ask what's different in the environment versus not. It would depend what our readout is and if it's transcriptomics or some other systems level phenotype for the different species.